Let's explore a plant cell's life cycle. Before meiosis begins, a cell undergoes interphase, a period of growth in which it continues functioning. Interphase has three phases, G1, S, and G2. In G1, the cell grows and does its job. In the S phase, the cell synthesizes DNA in the form of chromatin to prepare for meiosis. In G2, the cell continues to grow. Now the cell can be described as n equals 2, as it has two sets of chromosomes. This means that 2n is equal to 4, showing that with two sets of chromosomes, there are 4 individual chromosomes. Now we begin our journey through meiosis. The first stop is prophase 1. During prophase 1, the nuclear envelope disappears and spindle fibers form from microtubule clusters. Chromatin condenses to form chromosomes. Homologous chromosomes, ones which have genes in the same locations, align. In synapsis, homologous chromosomes are tightly connected by proteins. A tetrad is formed, composed of 4 chromatids. Then, a process called crossing over occurs. In crossing over, genetic information is exchanged between the homologous pairs. Next is metaphase 1. Spindle fibers begin to move chromosomes toward the center of the cell, known as the cell equator. In anaphase 1, homologous pairs are pulled apart by binding to opposite spindle fibers. However, sister chromatids stay together. The spindles attach to the chromosomes at the centromeres, which join the chromatids together. The next stage is telophase 1. A cell plate forms to divide the cell into two, an event called cytokinesis. The nuclear membrane reforms.
Now we've arrived at meiosis 2. Cells that go through meiosis 2 do so without replicating their DNA like in meiosis 1. The cells come directly from meiosis 1 and are haploid cells, meaning they have only one chromosome from each homologous pair. Each chromosome is made of 2 chromatids. The first stage in meiosis 2 is prophase 2. The spindle network appears while the nuclear membrane disappears. In metaphase 2, chromosomes line up along the cell equator by the work of spindle fibers. Next is anaphase 2. Spindle fibers separate sister chromatids and pull them to opposite poles of the cell. Proteins holding the sister chromatids together at the centromere are broken down. In telophase 2, nuclei in the cells reform and chromosomes revert to chromatin. Cytokinesis occurs via a cell plate and 4 haploid daughter cells are produced, each genetically different from the other. Here we end our journey through meiosis, with 4 non-identical plant cells that are called spores. Meiosis leads to the diversity we see in the world today. Crossing over in prophase 1 allows for the diversity in new cells, which is why each person has different DNA. Genetic variation ensures that species will survive environmental changes. Thanks for watching!